Hello everyone, welcome to this channel. My name is Ruben and today we're going to talk about the big ideas from ARKInvest from 2021, and this episode will be about part 14, multi-cancer screening. We are going to talk about the fact that liquid biopsies could prevent more cancer deaths than any medical intervention in history, and according to ARK's research, the convergence of innovative technologies has pushed the cost of multi-cancer screening down by 20-fold from $30,000 in 2015 to $1,500 today, and it should drop another 80% to $250 in 2025. And as a result of that, the multi-cancer screening market should scale to $150 billion in the United States. And a multi-cancer screening protocol could avert 66,000 cancer deaths per year in the United States. So that would be saving 1.4 million human life years. So what are the most exciting developments in cancer research?
We are starting to win the war on cancer. In fact, we lie at the intersection of three of the most exciting developments within cancer research. The first is cancer genomics. The genome is a composition of all the genetic information encoded by DNA in an organism. In cancers, changes in the DNA called mutations are what drives these cancers to go out of control. Around 10 years ago, I was part of the team at Johns Hopkins that first mapped the mutations of cancers. We did this first for colorectal, breast, pancreatic, and brain cancers. And since then, there have been over 90 projects in 70 countries all over the world working to understand the genetic basis of these diseases. Today, tens of thousands of cancers are understood down to exquisite molecular detail. The second revolution is precision medicine, also known as personalized medicine. Instead of one-size-fits-all methods to be able to treat cancers, there's a whole new class of drugs that are able to target cancers based on its unique genetic profile. Today, there are a host of these tailor-made drugs called targeted therapies available to physicians, even today, to be able to personalize their therapy for their patients, and many others are in development. The third exciting revolution is immunotherapy, and this is really exciting. Scientists have been able to leverage the immune system in the fight against cancer. For example, there's been ways where we find the off switches of cancer, and new drugs have been able to turn the immune system back on to be able to fight cancer. In addition, there are ways where you can take away immune cells from the body, train them, engineer them, put them back into the body to fight cancer.
Diagnosed early, cancer can be treated successfully. All solid tumors follow a predictable path from local and treatable to metastatic and lethal, providing the rationale for earlier detection. While ARK put this in a graph on the right, the distant metastatic cancers are the minority of the new cases, but the majority of deaths. And on the x-axis, they present the frequency of stages at diagnosis, and on the y-axis, we have resulting deaths after 5 years. You can focus on the purple. The metastatic cancers comprise just 17% of the new cases, but cause 55% of all cancer deaths over a 5-year period. If you look for green, this is regional, so only 24% of the cases cause 28% of deaths. And then we can move to the red, which is localized, which has a higher frequency, so 59% of cases only cause 17% of deaths. Based on a single blood test, multi-cancer screening can detect dozens of early-stage cancers. So along with somatic mutations, circulating proteins, and immune signatures, machine learning algorithms have surfaced DNA methylation as a novel and highly sensitive biomarker for earlier cancer detection. And the rapid cost decline of next-generation DNA sequencing is enabling liquid biopsies. And advancements in synthetic biology are helping clinicians to find faint signals of cancer in high-noise environments like the bloodstream. And on the right, they put this in a graph. It's called the combination of innovative technologies is driving down screening costs from 2015 to 2025. So on the x-axis, we have all these types of innovative technologies. And on the y-axis, they present the multi-cancer screening cost of goods sold in US dollars. While in 2015, it was like 30,000. And because of all this innovation, the cost is coming down to $1,500 in 2020, and ARK makes the estimation that this is going to be cheaper, like $250 in 2025. And that also creates an opportunity to invest in certain companies that are active in this field. So what are the current 3 ways to screen for cancer, and what are their disadvantages?
The key to cancer and curing cancer is early detection. And we intuitively know this. Finding cancer early results in better outcomes. And the numbers show this as well. For example, in ovarian cancer, if you detect cancer in stage 4, only 17% of the women survive at 5 years. However, if you're able to detect this cancer as early as stage 1, over 92% of women will survive. But the sad fact is, only 15% of women are detected in stage 1, whereas the vast majority, 17% of women, are detected in stages 3 and 4. We desperately need better detection mechanisms for cancers. The current best ways to screen cancer fall in one of 3 categories. First is medical procedures, which is like colonoscopy for colon cancer. Second is protein biomarkers, like PSA for prostate cancer. Or third, imaging techniques, such as mammography for breast cancer. Medical procedures are the gold standard. However, they are highly invasive and requires a large infrastructure to implement. Protein markers, while effective in some populations, are not very specific in some circumstances, resulting in high numbers of false positives, which then results in unnecessary workups and unnecessary procedures. Imaging methods, while useful in some populations, exposes patients to harmful radiation. In addition, it is not applicable to all patients. For example, mammography has problems in women with dense breasts. So what we need is a method that is non-invasive, that is light in infrastructure, that is highly specific, that also does not have false positives, does not use any radiation, and is applicable to large populations. Even more importantly, we need a method to be able to detect cancers before it's 100 million cells in size.
Thanks to rapidly declining costs, multi-cancer screening is approaching a reimbursable price point, while as population scale clinical utility data proliferates, ARK believes a $1,500 price tag will unlock the multi-cancer screening market for those aged 65 to 80. That's the age range in which the incidence of cancer peaks, and they also put this in a graph on the right. It's called the multi-cancer screening market, cost and reimbursements. So on the x-axis they present the patient age, and on the y-axis they have the incremental cost / life year gained in thousands of dollars. So multiple lines here. The green one is $2,000 test, the red one is $1,000 test, 1,500 is purple, and yellow is 1,200. And then we have this dotted line, also known as the willingness to pay, and indeed $1,500 will unlock the market. And as price drops below $1,000, nearly all age groups above 40 years could be screened for cancer effectively, and that will potentially save up to 1.4 million human life years in the United States alone. Routine blood-based multi-cancer screening combined with improvements in single cancer screening could prevent 40% of metastatic diagnosis and increase local regional diagnosis by 10%. While ARK makes an estimation, on the right we have earlier detection could lower cancer mortality. On the x-axis they make a comparison between motorcycle vehicle deaths and compare that with different kinds of age panels, and also make a distinction between a narrow panel and a broad panel. Well, a narrow panel seeks to detect a limited number of cancers, typically about a dozen, so approximately 12, and a broad panel, also known as pan-cancer screener panels, they aim to find as many as 50 different types of cancers. And on the y-axis they present the annual number of cancer deaths averted. And the point here is that even without improvements in cancer therapy, ARK estimates that earlier intervention could prevent 66,000 cancer deaths per year in the United States alone. A new way of early cancer detection screening is also known as a liquid biopsy, which is actually a blood-based test for early detection.
Central to our technology is a simple blood test. The blood circulatory system, while seemingly mundane, is essential for you to survive. providing oxygen and nutrients to your cells, and removing waste and carbon dioxide. Here's a key biological insight. Cancer cells grow and die faster than normal cells, and when they die, DNA is shed into the blood system. Since we know the signatures of these cancer cells from all the different cancer genome sequencing projects, we can look for those signals in the blood to be able to detect these cancers early. So instead of waiting for cancers to be large enough to cause symptoms, or for them to be dense enough to show up on imaging, or for them to be prominent enough for you to be able to visualize on medical procedures, we can start looking for cancers while they're relatively pretty small by looking for these small amounts of DNA in the blood. So let me tell you how we do this. First, like I said, we start off with a simple blood test. No radiation, no complicated equipment, a simple blood test. Then the blood is shipped to us, and what we do is extract the DNA out of it. While your body is mostly healthy cells, most of the DNA that's detected will be from healthy cells. However, there will be a small amount, less than 1%, that comes from the cancer cells. Then we use molecular biology methods to be able to enrich this DNA for areas of the genome which are known to be associated with cancer, based on the information from the cancer genomics projects. Then we're able to then put this DNA into DNA sequencing machines and to be able to digitize the DNA into A's, C's, T's, and G's, and having this final readout. Ultimately, we have an information of more than 5 billion billions of letters that output from this run. We then apply statistical and computational methods to be able to find the small signal that's present, indicative of the small amount of cancer DNA in the blood. So does this actually work in patients? Well, because there's no way of really sort of predicting right now which patients will get cancer, we use the next best population, cancers in remission, specifically lung cancer. The sad fact is, even with the best drugs that we have today, most lung cancers come back. The key then is to see whether we can be able to detect these recurrence of cancers earlier than standard methods. We just finished a major trial with Professor Charles Swanton at the University of College London examining this. Let me walk you through an example of one patient. Here's an example of one patient who undergoes surgery at time point 0 and then undergoes chemotherapy. Then the patient is under remission, where he is monitored using clinical exams and imaging methods. Around day 450, unfortunately, the cancer comes back. The question is, are we able to catch this earlier? During this whole time, we've been collecting blood serially to be able to measure the amount of ctDNA in the blood. So at the initial time point, as expected, there's a high level of cancer DNA in the blood. However, this goes away to 0 in subsequent time points and remains negligible after subsequent points. However, around day 340, we see the rise of cancer DNA in the blood. And eventually, it goes up higher for days 400 and 450. Here's the key, if you've missed it. At day 340, we see the rise in the cancer DNA in the blood. That means we are catching this cancer over 100 days earlier than traditional methods. This is 100 days earlier where you can give therapies, 100 days earlier where you can do surgical interventions, or even 100 days less for the cancer to grow, or 100 days less for resistance to occur. For some patients, this 100 days means the matter of life and death.
Multi-cancer screening and other genomic technologies are transforming oncology. Hereditary cancer testing can influence the onset and frequency of cancer screening. Multi-cancer screening can detect cancers in early and treatable stages. Single cancer screening plays a pivotal role in detecting several forms of cancer, so molecular prognostic testing can help pathologists differentiate between aggressive and indolent cancers, reducing overtreatment. So somatic and minimal residual disease testing, also known as MRD, enables oncologists to optimize surgical resection, identify precision therapies, and monitor for cancer recurrence. Sizing the opportunity. What are we talking about? While a 1500 unit price could unlock the market, a $1,000 unit price could transform the cancer mortality curve. On the right, they put this in a graph. It's called the multi-cancer screening is potentially one of the largest genomic markets. The x-axis represents the reimbursement price in US dollars based on a logarithmic scale, and the y-axis represents total addressable market in US billions of dollars. So the conversions between and among these innovative technologies has pushed the cost of this multi-cancer screening down by 20-fold from $30,000 in 2015 to $1,500 today. And according to ARK's research, this could drop another 80% to $250 in 2025. So if fully adopted, multi-cancer screening should scale to a $150 billion market in the United States alone. Thank you everyone for watching this video. I really hope you enjoyed it and that you actually learned something, and see you on the next part.